So hello, everybody. Thanks so much for sticking around for this late talk in the day. Today, I'm going to give you a brief overview on a Python package, an open source one we developed called EUGENE, a Python toolkit for predictive analysis of regulatory sequences. So the biological focus of my short talk today is on gene regulation, specifically the process of transcription or the generation of these messenger RNA molecules that serve as intermediate functional gene products. And we can define the regulatory genome then as everything that influences the timing, location, and quantity of gene expression. And building maps that really tell us how this regulatory function is encoded in our DNA is one of the fundamental challenges of genomics research. And more specifically, we'd like to build quantitative and context-specific maps that tell us not only where functional regulatory DNA exists, but also the effect of variation in that sequence at nucleotide resolution. But this space is kind of currently intractable to experimentally resolve. So what we do as computational biologists is build models, and a particular modeling strategy that has become, gained a lot of attention in the last 10 years or so has been to predict functional genomics data from DNA sequence alone. And this allows us to ask many diverse questions and distinct questions about how the transcription is regulated by information encoded solely in DNA. But this explosive growth is not without technical challenges, as always. Building these sequence-based deep learners involves many steps, each with specifics nuances that are specific to genomic sequences. And though conceptually a lot of these are pretty straightforward, many of them are not often handled well by generic machine learning platforms. And so this in turn has led to the development of several tools and code bases that unfortunately are often compatible with a single model or model type and don't work well with each other. And so by themselves, all these tools and code are not comprehensive enough to run this full workflow end to end. And this in turn causes several problems for both tool users and developers. It hurts research and reuse, it hurts reuse and reproducibility, leading to less trust in our models. It creates this disconnect between those who generate the data and the methods and models that can extract insights from this data, and it just plain slows down research. So all this really motivated us to develop an open source Python package called EUGENE, or elucidating the utility of genomic elements with neural nets. In this presentation, I'll just briefly highlight the hallmarks for each of the major stages of the workflow illustrated on this slide. But if you're interested in more of the details, you can check out our preprint, EUGENE's GitHub, and the documentation pages. So for data handling in EUGENE, we built standalone Python packages called SeqData and SeqPro that very flexibly read files from on-disk formats into X-ray datasets that are backed by these ZAR stores. We then implement on top of this many functions that can be called directly on these X-ray datasets to perform, my cursor's showing, it doesn't matter, one-hot encoding of sequences really fast, target normalization, data visualization, and conversion of this data format into formats that are ingestible by deep learning frameworks like PyTorch. For model building and training, EUGENE serves as a nice bridge between the PyTorch deep learning library and these sequence-based models, offering a growing zoo of customizable model architectures that can be instantiated with single function calls, several methods for initializing those models, and a PyTorch Lightning interface for training models and using them for inference. Finally, for model interpretation, EUGENE provides a suite of analysis tools commonly used for these sequence-based deep learners, including functionality for feature attribution, global importance analysis, and sequence evolution, all implemented in another standalone Python package called SeqExplainer. In practice, these features make EUGENE a great tool for improving reproducibility and reuse, which we highlight in 3 diverse use cases on our preprint, and many more in our dedicated use cases GitHub repository here. By aggregating all these methods and models together in one tool, one modular, easily accessible, and user-friendly tool, EUGENE lowers the barrier for both data generators and computational scientists to access these methods. And finally, EUGENE shifts the focus from reinventing the wheel to innovating it, allowing users and developers to more readily move their research forward. And just in summary, hope I've convinced you that EUGENE addresses these unmet need for a simplified and comprehensive framework for doing this. And we do envision, I'll say quickly, something very similar to those single cell enthusiasts out there in the SCVerse framework for doing this in the future. And with that, I'd like to thank my wonderful collaborators and funding sources, and also thank BOSC and ISMB for giving me the chance to present my work. A couple links here. If you're interested in chatting more, please come find me.